To begin with some disclosures, potential conflicts of interest, and any sources of potential bias, you should know that I've been on creatine since about 1992. I don't know that that will influence the results of this talk. I've published and presented a fair bit about creatine and other supplements. I've been funded by the NIH, various foundations, universities, and occasionally companies. I'm currently not funded by anyone. Occasionally, someone thinks I deserve some sort of honoraria for lecturing about creatine, and I'm on the Scientific Advisory Board for ALSChem, but to be fair, I did study and publish about creatine for 20 years before that happened. And I'll just do my absolute best, thank you, to give you a fair and unbiased perspective on the literature related to creatine, supplementation, and brain health. So over the past few years, I've given a similar lecture, and I've gotten in the habit of closing with a slide like this, where I say, now you know what we have, here's what I think is missing, here's what I think is desperately needed for research. So over the past 2 years, what I've been pointing out is that in terms of creatine supplementation and brain creatine levels, we have a desperate need for dose-response studies. We have a desperate need for regional differences studies, reliability studies. We really need to understand more about muscle and brain simultaneous responses to supplementation. And it seemed for a period of time that some labs measured total creatine, some measured phosphocreatine, but very few measured both, and that's the limitation of the literature. And in terms of brain performance, it seems like there's two groups of researchers, those who measure cognitive processing and those who measure metabolic changes in the brain, and we need to bring those two groups together. There's very little evidence or research in brain performance as it directly pertains to sport, and we need to do a much better job highlighting the effects of creatine supplementation on concussion and mild traumatic brain injury. So that's been my closing slide for the past 2 years or so. You'll get the most recently updated version at the end of today's presentation. So the big bang of creatine supplementation was really Rogers' 1992 paper. This has been cited over 1,800 times, but there's another important paper that is not as widely cited. It's been cited a little over 300 times, and it's the paper from Deschent and colleagues published in 1999. And to compare these two seminal works, from Rogers' paper, we learned that oral supplementation could in fact increase skeletal muscle creatine. We learned that on average this is about a 20% increase. Those of us with low initial muscle creatine have a larger increase in response to supplementation, and that exercise increases uptake. From the Deschent paper, we learned that oral creatine supplementation could in fact increase brain creatine, which is a remarkable finding. The increase is smaller, 10% in this paper, but possibly even smaller than that. And we learned that there are regional differences in uptake. What's important from the Deschent paper is that we realized that there are very large differences between muscle and brain. So in considering the differences between the muscle response to creatine and the brain response to creatine supplements, most of the creatine in your body is stored in your skeletal muscle. Only about 5% or so is in your brain. This is important because there is a separation of the sites of creatine synthesis and the sites of creatine storage and utilization when it comes to skeletal muscle. This makes skeletal muscle really designed to take up exogenous creatine. But the brain has its own metabolic machinery to synthesize creatine, and by nature it would make brain very resistant to take up exogenous creatine. I could put you on a vegetarian diet today, and in 2 or 3 weeks you would have drastically reduced muscle creatine. As far as I know, there's no effect of a low creatine diet on brain creatine levels. Now, don't confuse this to mean that there's no effect of a low creatine diet on brain function. This refers to if I compare brain creatine levels in vegetarians and meat eaters, I get the same values. There is some evidence now that I'll talk about in a moment that a chronically low creatine diet actually alters brain function. Under extreme circumstances, muscle creatine decreases with low levels of physical activity. What I'm thinking of here are the classic works of a great exercise physiologist, Duncan McDougall, who used limb immobilization to show rapid reductions in skeletal muscle creatine. I have no idea if extremely low levels of physical activity have any effect on your brain creatine levels. I also have no idea if low levels of brain activity have an effect on your levels of brain creatine. I know that your exercise physiology textbooks tend to say that exercise training increases skeletal muscle creatine levels. They're incorrect. I'm right. I don't think that muscle creatine changes at all in response to sprint exercise training. More importantly, I really don't have any idea if brain exercise or physical activity can increase your brain levels of creatine. I'm confident when I see biopsy data from the different people in this room that the brain creatine values are stable, because I know those muscle creatine levels are stable day to day. I know they're stable at rest. I know the reproducibility of muscle creatine assessments. I have much less confidence in the brain levels, but I'm renewed and excited because we're going to hear from Jed, who's going to present some research of his and Craig Sales after my presentation about the reliability and the variability in brain creatine levels. So I'm very excited for that talk. So that's kind of the basic quick background here. The first question we need to answer is perhaps phrased here a bit too simplistically: can we increase brain creatine with creatine supplementation? And you've seen a figure like this several times at this meeting already, with creatine synthesis on the left, and the image on the right is kind of used to teach that the transporter necessary for creatine transport between brain cells is there, but the lack of this transporter on the astrocytic feet means you can't supplement creatine and have it taken up by the brain. I don't think this is a yes or no type of situation, and I wish people would stop explaining it that way. When I hear people say that, you know, the enzyme isn't available, the transporter is not available on the astrocytic seat, I refer them to these data, which some of you may be familiar with if you're as old as I am. I understand that these are from creatine transporter deficiencies or creatine enzyme deficiencies, but if you look at these data, in these children, they show brain creatine uptake. And I understand the timeline is different. Those are not days on the x-axis, those are months. But in some cases with these kids, you're getting 50 to 100% replacement of brain creatine with supplementation. So it's not that it can't get into the brain, it's that we have to be very intentional about the circumstance where we are supplementing creatine and what sort of response we should expect. Now, what sort of response should we expect? By my count, there's about 13 studies of healthy adults who have been supplemented with creatine, and we had pre and post measurements of brain creatine. There are more studies here in clinical populations, particularly in patients suffering from depression, which Perry will talk about after my presentation. In my interpretation of the literature, 10 out of the 13 studies show with reasonable supplementation doses, there's an increase in brain creatine levels, total creatine or phosphocreatine. There's one fantastic paper from Bruno's lab in Brazil that has simultaneous measurements of muscle and brain creatine levels, and it does a nice job demonstrating that in terms of skeletal muscle, everybody increases their muscle creatine levels in response to supplementation. It would be a small, a medium, or a large increase, but everybody goes up. When it comes to brain creatine, it appears that some people don't respond at all, whereas others have a small positive increase. And you can see similar results if you go back to the original Deschent paper, which the top table shows that there is a range of brain creatine increases from 3 up to 13%, and then some regional differences between 5 and 8.7%. So it's not the same as skeletal muscle at all. The increase on an absolute basis is much smaller, and we're dealing with regional differences as well. This is another paper that I'm fond of sharing from Bruno's lab in Brazil. And what we have here is a nice demonstration of the variability in the response to creatine supplementation when it comes to the different regions of the brain. So there's 3 different regions of the brain. There's pre-post supplementation in the creatine group and in the placebo group, but if you look at that red rectangle, I have hidden which is the creatine group and which is the placebo group. So I encourage you to pick a panel, A, B, or C, pick a side, left or right. So let's say we look at the middle panel and the right side. Can you say with 100% confidence that that is either the placebo group or the creatine group? Right, I see extreme increases and extreme decreases. If we look at the bottom panel on the left, I see similar sized increases, but I see the same number of increases as I do decreases. Right, so there's some differences here between skeletal muscle creatine uptake such that I can't really know what to expect in terms of changes in response to supplementation. And I'm not going to tell you which is the placebo group or the creatine group. So what does the literature tell us? Can we increase brain creatine with creatine supplementation? Yes, but let's place things in the proper context. We have to respect the variability in the response if we're going to properly interpret these data. We really have to remember as we go forward with these studies that skeletal muscle and brain are not the same tissues at all. This is important because I do have some biases. I'm a muscle physiologist, exercise scientist, right? I had to learn brain physiology very, very late in my career. So this is something that I'm constantly working hard to learn about. So if I supplement with creatine and I do increase my brain creatine levels, can I alter my brain function? Can I improve my cognitive processing abilities? This is something that's been discussed a number of different ways, and I'll try to present the results of these data from several different perspectives. There's a number of narrative reviews on the effects of creatine supplementation on cognitive processing and brain health. There are also at this point several systematic reviews and meta-analyses, so we can look at the effect size from a quantitative standpoint, because I think there was some criticism that there were only narrative reviews available, but now we have qualitative and quantitative assessments of cognitive processing outcomes. So again, I'm aware there are more studies here. I'm excluding the clinical populations. I'm going to let Perry talk about creatine supplementation in depression. I count 18 studies of healthy adults ingesting loading doses or traditional doses of creatine monohydrate supplements. 14 of these studies showed an improvement in cognitive performance. 4 of them showed no effect. Now, again, there's a number of different reviews here. I think Darren's review in sports medicine is probably the most current. As usual, it's also the largest. And all of these reviews tend to highlight the same thing. These studies are very difficult to compare, right? Some of the 18 studies were focused on young adults. Some of them involved older adults. In some cases, they were vegetarians. In some cases, they were meat eaters. A large difference between these studies is that some of these studies examined the effects of creatine supplementation on cognitive processing under resting conditions, whereas other studies had some sort of stress imposed on the research subjects. So this was a mentally fatiguing task, or a physically fatiguing task, or both, or sleep deprivation. In addition to that, the outcome measurements are wildly different, right? If I measure strength, and Darren measures strength, and Rick and Mark measure strength all over the United States, we all measured something very similar, and we understand that we were measuring the same thing, muscle strength. But when it comes to the cognitive processing outcomes, there is some disagreement with how best to assess the general, cognitive processing outcome measure. So there's different outcomes. There's also different dosing strategies, different supplementation duration. People have used this as a criticism of the literature. I think this is fantastic, because with all this noise, with this mess, Overwhelmingly, these are positive effects. And we're talking about a nutrient, a single nutrient improving cognitive processing outcomes. That's fantastic that we can detect that at all. And the fact that we can detect it consistently in groups who are relatively healthy, not brain creatine deprived in any way, with all of these differences in methods, I find that to be actually quite inspiring. So if you were to ask me flat out, is there an improvement in cognitive processing in adults with creatine supplementation, I would say absolutely, that's what the literature supports. Now, the quantitative data. There's a few systematic reviews, a few meta-analyses now. You know, we could debate at what point one gets to conduct a meta-analysis. Do we have to wait for 50 studies or 100 studies? So these are small systematic reviews and metas, but I think they're valuable. The problem is that they often don't have that many outcome measurements in common. The thing that is measured most frequently in these studies is memory. So here's one of the forest plots from the NICE meta published in 2024 in Frontiers in Nutrition, showing that in approximately 500 subjects in about 16 randomized controlled trials, there is indeed a statistically significant improvement in memory. And I'll say again, to be able to measure an improvement in memory with a single nutrient is really fantastic. It really is. Now that's two ways to describe the data, the narrative reviews and the quantitative reviews. And then there's one more way to examine this, and you heard a lot about dietary creatine from Sergey, but I want to point out one article from Marco Machado, which Darren is actually a co-author on. And this made the connection between dietary creatine, so it's not a supplementation study, but it shows that there's a statistically significant and I think clinically relevant relationship between dietary creatine intake, which was very low in these individuals, and memory. So at this point in time, we have supplementation studies with reasonable doses showing, yes, we can alter brain energetics, and we have supplementation studies in healthy individuals with reasonable doses showing in narrative reviews and in meta-analytic reviews an improvement in memory. And we also have it from kind of an epidemiological natural history type of approach where people's dietary behaviors when it comes to creatine are having an impact on cognitive processing. Now, the big question I get whenever I do one of these lectures is what is the proper dose? And the answer simply is we don't know, right? We have all of these biopsy studies. There has been an amazing work, 30 years worth of work on establishing an appropriate dosing strategy to increase skeletal muscle creatine. This really doesn't exist for brain creatine yet. Our best guess is to attempt something similar to muscle creatine loading, although there are some clinical studies, like for instance Huntington's disease, where they used much higher doses. We don't really have any evidence to say you should absolutely be taking 30 or 40 grams per day. So the safest thing that we know about, the most, when it comes to skeletal muscle, the best data are supporting a traditional loading protocol of approximately 20 grams per day. We have a desperate need for dose response studies. We tend not to do these in nutrition and exercise research. We do these all the time in pharmacy research, but we tend not to do these. The only paper that I'm aware of that really tried to get at this was, again, from Darren, who's been incredibly productive in this space, where they used two different dosing strategies, kind of a modest dose and then a much higher dose, and then they were unable to generate a beneficial effect, and then, of course, had no difference between the dosing strategies. But we need a lot more work in this area. Now, why aren't we doing studies in this area? Probably because it's phenomenally expensive, right? So if you need an MRI, if you need to do brain spectroscopy, you need access to a magnet, okay? And then that magnet has to be a dedicated research magnet, otherwise you're competing with diagnostic radiology, and you're not going to get much magnet time. If we considered that a single brain creatine measurement was $500 US dollars. So pre-post supplementation, 500, And if I'm asking to do brain pre-post, muscle pre-post, that's $2,000 per subject. And if I want to do multiple doses, right, 6, 7, 8, 10 times $500. If I want to do a washout period, which is also completely unknown, it becomes very expensive, very quick. And I think lack of access to the equipment and lack of funding in this area is really what's not allowing this question to be answered as fast as I would like it. So I'm going to make kind of an odd suggestion here. I think that it's possible we could answer some of the questions to do with dosing strategies using case studies. Now, when I teach my students that case studies are generally the lowest form of science when it comes to research design. And that's because when it comes to case studies, the things that wind up coming across my desk are things like this, right? Yeah, it has to be the creatine. So in our review in sports medicine, Mark and I and the late Priscilla Clarkson reviewed some of the creatine supplementation case studies involving rhabdomyolysis, and the number of mitigating circumstances were incredible. Yet creatine grabbed the headline. Creatine was always featured in the title of the article. I don't mean retrospective case studies. I mean well-planned, prospective, moving forward case studies where we have exquisite control over the subject's physical activity. We can test for cardiorespiratory fitness, muscular fitness, body composition. We can control their diets, and we can do multiple doses, multiple dosing strategies, and even washout periods for a reasonable amount of money. I get it, an N of 1 is not that impressive, but I think from a foundational perspective, these will be incredibly informative and will push the literature forward. And maybe this will be the foundation for us to be able to do randomized controlled trial dosing studies. So not that type of case study, the higher quality kind. No disrespect to those investigators. I understand clinicians, someone stumbles into an emergency room, you get the best medical history you can, and then whatever they say, you blame creatine. Hey, is there an acute effect? You saw a nice summary from Ali. This is my absolute favorite paper. I think this is the paper of the year because it's reinvigorated the way I'm thinking about creatine supplementation and cognitive processing outcomes. Chad did a nice job reminding us that an acute dose makes absolutely no sense from a skeletal muscle standpoint, but no one was really thinking about it from a cognitive standpoint. And we had suggested a model, which you'll see in a few slides, about when someone is faced with a large enough acute stress that you need some sort of a safety net for that acute stress. And it looks like creatine could really play an important role here. We have evidence from Ali's study on improvements in brain energetics, improvements in cognitive processing. This is in using a reasonable dose of creatine combined with sleep deprivation. I think this has also opened up a whole different avenue of thinking for especially athletic populations. And thank you to Chad for bringing up that Cook paper on rugby players, because that's one of the most overlooked papers in this body of literature. I want to address the mental fatigue issue if I could, because we casually say things like, I'm going to induce mental fatigue, as if we know how to do that as well as we know how to induce physical fatigue, right? I can make your muscles tired. I'm good at that. Inducing mental fatigue. The best paper, I think the gold standard paper, is the paper from Van Cutsem. This is from Romaine's lab. Most of you, if you're like me, everything you know about the interactions between nutrition and the brain, like carbohydrate and caffeine in the brain, it comes from Romaine's lab. This was a nicely done short-term study where they were able to show that creatine improves hand grip endurance as well as Stroop task accuracy following a mentally fatiguing task. Now, what was the mentally fatiguing task? Because I don't think we all want to get into the sleep deprivation business. Some of us are interested in mentally fatiguing tasks because that's a logical extension to sport or to other types of physically demanding and mentally demanding types of stressors. So they use the Stroop task as their mentally demanding task. So if you haven't thought about the Stroop test in a while, play along. We're looking for speed and accuracy. Okay? Read the color of the word and time yourself. So if we go across the top row, ready? We're going green, red, blue. Row 2, yellow, blue, black. Row 3, red, green, blue. See? Now, in Romaine's study, they did the Stroop test for 90 minutes straight. Everybody ready? Okay, so when I see these studies where they say we had someone count backwards from 100 and stood on one leg and they induced mental fatigue, no, you didn't. Okay, that's one of the best labs in the world and really understands the interactions between nutrition and brain function. So we have to be very intentional about how we're fatiguing the brain. It's easy to induce cognitive impairments with sleep deprivation, and we've shown on several occasions, acutely and more chronically, that you can rescue people from that decline in cognitive processing with creatine monohydrate, which is fantastic. But how does this relate to people in real environments who are perhaps not sleep deprived for 24 or 36 hours? 90 minutes, can you imagine? When I ask you to think what type of athlete would benefit from creatine supplements, perhaps acute supplements, because we have mentally and physically fatigued them, what do you picture? What's the task, right? Is it an endurance task that's incredibly repetitive? Will that athlete receive some sort of mental benefit from perhaps pre-race creatine supplementation? To be clear, when we're calling this an endurance event, but he's running at over 13 miles per hour for 26 miles straight, so that's a sprint for, that's even more than a sprint for most of us. Is it being perfect, being precise amidst all of this chaos at the end of a game? Is that the athlete who would benefit from an anti-mental fatigue type of supplement? Is it someone who needs to be perfect in terms of maximal output at the end of several challenges of maximal output? Is it a matter of life and death if you're standing in front of Mr. Pereira? Is it a muscular endurance task in a fatigued athlete? Is that where we need this improvement in brain energetics? Or is it, like that rugby study, is it team sport specific? Is it a combination of sprinting, endurance, fatigue, where you need to be mentally at your best? Is that causing the mental fatigue, like that Stroop test, that we could rescue that person with an acute dose of creatine, right? None of these questions have been answered. The creatine brain function literature when it comes to sports science has been almost completely ignored. We're doing our best work in the lab with computer-based cognitive processing tests, which is lovely. We understand the reliability and the validity, where we're making people do the Stroop test for 90 minutes straight. I can't generalize that too much. So there's so much great work to be done here in sports science, and I hope we'll consider that for this sports session on the last day. It could be a population we're not thinking about at all. And I think that's what your paper really opened my eyes to, again, is that we're not thinking broadly enough, right? And the military applications, I think, are endless in terms of physical fatigue, physically and mentally demanding tasks. Okay. Do improved cerebral energetics / cognitive processing via creatine supplementation during fatiguing sport matter? That's focus right there. I don't have an answer to that question. We just have that one rugby study that never really seemed to be followed up. And I would encourage you on your travels home to think about a lot of the fun projects that could be done in this space. All right, what else do I have for you? I want to make a few comments about the strengths and the weaknesses of this literature. And the most recent review, and I think everyone who's interested in the cognitive processing end of things should read Terry McMorris's review, where he kind of encourages us to exercise caution and not speak beyond the data, but admits, yeah, there's a significant improvement in cognitive processing here, especially on memory. It's funny to see Terry be so conservative because he really was foundational in this entire field, right? He has the only study of frail elders, and these were not just healthy 62-year-olds, these were frail elders, and he also started all of the sleep deprivation work. And the reason Terry has been so important in this field is because he's not like me. He's not an exercise physiologist. He's more of an exercise psychologist, exercise physiologist. He's a brain person. So I've been on my way here reading an awful lot about how mental fatigue is important in terms of sporting performance and what actually are the causes of mental fatigue and what type of exercise induces these changes. And it's an extremely complicated body of literature. This is an excellent review to start with. It's humbling and lets you know how much you don't know about a specific aspect of sport performance that's very much tied to creatine supplementation. So as always, thank you to Terry for being an inspiration in this space. Okay, so we've proposed this model, and I think Darren showed this image the other day. We believe there are benefits to creatine supplementation, to increasing brain creatine, especially if you're going to be faced with some sort of acute or chronic stress. The chronic stress would be something like depression or perhaps aging. And acute stress might be something like sleep deprivation or some sort of mentally demanding and physically demanding type of task. There's one situation that to me bridges both the acute stress and the chronic stress, where having a reserve of creatine in your brain could be incredibly important, and that's mild traumatic brain injury. So I'm going to walk us through the story of concussion, how it relates to creatine metabolism, what we know about creatine supplementation, and then finish up with some suggestions about what to do next. I think you've seen this image, or some of you have. This is the neurometabolic cascade of a mild traumatic brain injury, right? It's not simple. In red is where concussion overlaps with creatine biology. And what you have is some sort of a mild traumatic brain injury. There's large-scale membrane depolarization. There's increased energy demands, but there is decreased brain creatine. Right away, you should be thinking, this seems like an opportunity for a nutritional intervention, and I think you'd be correct. There is reduced blood flow, increased oxidative stress, increased intracellular calcium. Essentially, everything that could possibly go wrong does, but so much of it overlaps with creatine biology, right? So on paper, in theory, creatine looks like an excellent intervention to either decrease the damage from a mild traumatic brain injury or perhaps enhance recovery from. That's speaking theoretically. We have some animal research, and the results are quite eye-opening and very, very positive. With an animal model, and I rarely, if ever, discuss an animal model, but we're talking about brain injury here, we can induce hypoxic brain damage or traumatic brain damage in an animal. My IRB won't let me induce traumatic brain injuries in people. So you don't know who's going to get a concussion or when you can intervene. So the animal models are quite important. 36% and 50% decrease in damage in mice and rats, respectively. Protection appears to be related to mitochondrial bioenergetics. Mitochondrial membrane potential significantly increased. Intramitochondrial levels of Rawson and calcium significantly decreased. ATP levels maintained. On all of the fronts from that previous slide, which was in theory, we see creatine improving outcomes to traumatic brain injury in an animal model. Do we have any human data? We have two types of human data that I'll share with you. These are traumatic brain injuries, thank you, in children. These are hospitalized children. They're very sick. And they were given creatine supplements and followed for 6 months. The graphs are for remarkable improvements in headache, dizziness, and fatigue. This is an open-label study. You know, if it's not a randomized controlled, double-blind, placebo-controlled trial, I usually don't mention it, but these kids are in need of help, right? They're sick. Positive effects for headache, dizziness, fatigue, but also post-traumatic amnesia, days of intubation. This is a hospital. They have more data than they know what to do with. Communication, personality, behavior, cognitive function. The creatine supplementation improved all of these different outcomes. It's not perfect. It's not double-blind, randomized, placebo-controlled trial, but it's very, very promising. And then there's some unique data from this group who has access to retired National Football League players. If you're not familiar with this lab, Michael Alasko, there's a lot of studies related to the brain and retired National Football League players. And they were able to convince me with this particular study that there's late life derangements in energy metabolism in people who have had repeated head trauma. So their players who are still symptomatic years after retirement have reduced brain creatine related to the frequency of their head injuries. So that's a really interesting way to look at it. It's one study, but it is the most interesting population. So if I put this all together, I get a lot of really lovely circumstantial evidence, which I usually don't, get on my soapbox about, but I think it paints a really incredible picture. Theoretically, all of the things that go wrong with a mild traumatic brain injury really look like they could be somewhat mitigated by increasing brain creatine levels, which we know decrease with a brain injury. The National Football League players, persistent long-term changes to cognitive, behavioral, and mood symptoms are related to reduced brain creatine years after retirement, indicating some sort of later life derangement of energy metabolism. The animal data in rats and mice, I'm aware that rats and mice, when it comes to brain creatine, respond to a much larger extent than humans, right? So this is not perfect, but it's still very, very compelling, particularly the large reduction in damage. And then the human data from the Children's Hospital open label trials showing remarkable improvements in children giving creatine supplements. How can we make some sense of these data? I'm going to be very practical here. With creatine supplements, I have documented muscular benefits, an inexpensive, widely available supplement with an incredibly strong safety profile. Is encouraging supplementation to reduce damage or enhance recovery from mild traumatic brain injury premature? I would say encouraging supplementation for high-risk populations is actually the most prudent thing you could do. I don't think it makes any sense at this point in time to say we should wait for more information with a high-risk population. So I've marked this up a little bit for you. Where do I think we go moving forward? I think we still need some dose response studies. We have to figure out the different dose between muscle and brain. I think we can accomplish a lot with really good prospective case studies. There are still very few simultaneous brain and muscle studies, although we're going to learn a lot about regional differences, reliability, and individual variation from Jed's presentation. A lot of labs seem now able to measure total creatine and phosphocreatine, so we're getting a more complete picture. In terms of brain performance, there are still very few labs who measure cognitive processing and brain metabolism, so we need more papers there. The sports performance or the military applications are wide open, and the concussion, traumatic brain injury, I think we could use some really good case studies there. One thing that we didn't really talk about is the fact that not every athlete gets a traumatic brain injury. I think we should be studying subconcussive forces as well, because there are a lot of athletes who are hit in the head a lot of times, but don't necessarily get a brain injury. So I'll share with you this network map of some of the people in this room and in this field. I'll say thank you to Alice Kem for the kind invitation, and thank you to Rick, and thank you to all my collaborators who were all connected in one way or another through this very interesting nutrient. Thank you.
much.